Hello everyone, welcome to our channel Concepts of Biology. In this video, we will be studying how the proteins are transported to their designated places. Whenever we talk about the proteins, first of all, the protein synthesis involves the two processes, transcription and translation. In transcription, the mRNA is formed inside the nucleus, and in translation, this mRNA results into formation of the protein via translation by attaching themselves to the ribosomes where the process of translation occurs. Where the protein is having some target sequence attached to it which targets the proteins to their designated places. You can see here the nascent protein is having some signal sequence. Here the ER signal sequence is present in this protein. So this protein is being targeted to the endoplasmic reticulum. First of all, the mature cytoplasmic protein is having its own target sequence. It either enters the plasmic reticulum or to some other organelle present inside this cytoplasm. The ribosomes are mainly of two types. One are attached to the surface of the endoplasmic reticulum. The other are present free in the cytoplasm. The ribosomes which are present free in the cytoplasm results into the formation of mature cytoplasmic proteins which will be having some specific uptake targeting sequence which will tell it whether it has to go into the mitochondria, chloroplast, peroxisome or back into the nucleus. And the other type of ribosomes which are attached to the ER synthesize specific type of ER signal sequence. Let's proceed forward. How this sorting takes place. First of all, whenever a new protein is synthesized, it is seen that whether this protein is having the end terminus of the protein is hydrophobic or not. If it is hydrophobic, then it will enter inside the rough endoplasmic reticulum, and if it is not hydrophobic, then it will be forming the cytoplasmic type of proteins that will enter either of the organelles, the nucleus, mitochondria, chloroplast or peroxisome. If a protein enters this ER, then it will either have this KDES sequence at its C terminus. The C terminus of this proteins will be having the KDES sequence. This KDES sequence will restrict the proteins into the ER and will not allow this the transport of proteins into the Golgi. If this sequence is not present, the proteins will be transported to the Golgi. So, whenever protein present is transported to the Golgi, if it is not, it is seen that it is having a alpha helix sequence or not. If it is having the alpha helix sequence, then it will undergo the retrograde trafficking that we'll be studying in the upcoming slides. If it is having the alpha helix sequence, then it will undergo the regulated secretion, constitutive secretion, or attaches with the lysosomes to form the endosomes. The transport of the proteins, the extracellular of the transport of the proteins, we have already studied in our previous video. In that video, we have also studied about this retrograde transport of the proteins. I'll also explain this in this slide. This also. Let's see how this occurs. Here specifically the ER specific transport of the ER specific proteins is shown in this slide. You can see in this diagram, you can see if the protein was having this KDEL sequence, then it will undergo the retrograde transport. That means the Golgi will be having the COP1 type of, it will be forming the COP1 type of vesicles and will return back to the ER. This is called as the retrograde trafficking or retrograde transport of the proteins. When the transport of the proteins takes place from ER to Golgi and then undergoes secretory, constitutive or neglected secretion, or will enter the lysosomes, thus forming the endosomes. This type of transport is called as the anterograde transport. Here, the COP2 vesicles are formed. The vesicles leaving the ER are having the COP2 type of goal. See this coating of the vesicles. Then some cladrine type of, third type of coating, that is cladrine coating, is also discussed. Here the vesicles which leave the Golgi apparatus and joins this lysosomes, this forming the endosomes, or this you can also say the the vesicles appearing from this plasma membrane by the endocytosis are also having the cladrine type of coating. The one arising from the Golgi are having the cladrine AP1 type of coating, and the one which are arising from plasma membrane are having the cladrine AP2 type of coating. Let's discuss it in a little detail. The COP2 vesicles mediate the cargo transport from the ER to cis Golgi apparatus. Here you can see the COP2 vesicles. These blue colored vesicles are the COP2 vesicles. These are leaving the ER and then getting fused with this cis Golgi compartments. These are having the COP2 type of protein coatings, and the one which are undergoing this retrograde transport from the ER, from the Golgi to the ER, are having the COP1 type of vesicles. The other ones which are leaving this Golgi and attaching this lysosomes or arising from the plasma membranes, these are cladrine coated type of vesicles. Then some other type of vesicles which are called as retromites are also present here. The vesicles which attach with endosomes and the sperm enters into the vacuoles are called as this retromers. Then all these transport across these membranes, the transport of the vesicles across the membranes from ER to this Golgi, and from Golgi to the plasma membrane requires some special type of you can say the energy particles that are called as RAB GTPases. Then that's all for today. Thanks. Please like, share and comment. Also subscribe to this channel.